Hello everyone, I am Professor Yeon-soo. I will talk about the foreign animal infectious diseases. The foreign means in Korean side. In Korea, the the diseases that I am going to talk about is not yet outbreak. The first, I will talk about the Pestipity ruminants. So we call that small ruminants the rinderpest. The rinderpest-like diseases, but the outbreaks occurred in small ruminants. The background. The Pestipity, the ruminants is also known as goat plague, kata, syndrome of stomatitis pneumoenteritis. The stomatitis is the oral infection, and the pneumo is a respiratory, enteritis is an intestine inflammation, or even ovine in rinderpest. PPR is an OR listed disease and must be reported to the OR according to the the animal health code. It is an important infectious viral disease of the domestic and wild small ruminants. The Pestipity ruminants virus belongs to the genus Morbillivirus of the family Paramyxoviridae, closely related to rinderpest disease, rinderpest virus. It is characterized by the severe morbidity and mortality rates. In areas where the disease is endemic, the mortality rate may be lower, but the disease has a more insidious impact on flock productivity. This is the Pestipity ruminant virus, PPRV. The genus Morbillivirus and the subfamily Paramyxoviridae, the family Paramyxoviridae, order Mononegavirales. The virus has a membrane envelope, and a nuclear capsid which contains a negative sense single-stranded RNA genome. The official name of this virus was changed in the 2016 to small ruminant morbillivirus. However, it is still commonly known as PPRV by people working in the field. Actually, we are easily remember the PPRV. The molecular epidemiology of PPRV based on the F gene, the N gene, or the H gene sequence and has defined the existence of four lineages. Four lineages it has. The PPRV is closely related to the rinderpest virus. As you already knew, the rinderpest is declared disappear. The disappearance is declared. That causes disease in cattle, buffalo, sheep, goats, and wild ruminants. The measles virus I'd like to talk about the family viruses. The family viruses belong to the Paramyxoviridae, the morbilliviruses. The first is a measles virus. The measles virus is one of the most recognized human pathogens in the morbillivirus genus. We all get vaccinated when we are very, very young, maybe before toddler stages. We all the three more times we got vaccinated against the measles. Causes measles, a highly contagious disease marked by the fever, cough, rash, and in severe cases, complications like pneumonia and encephalitis. The vaccination has been highly effective in reducing measles cases, though outbreaks still occur, especially in areas with low vaccination rates. And the very famous rinderpest virus, the RPV, known as the cattle plague. Rinderpest virus caused a highly infectious disease in cattle and other ruminants, leading to high mortality. The rinderpest was one of the deadliest livestock diseases before, with symptoms including fever, diarrhea, and respiratory distress. Through extensive vaccination efforts, rinderpest was declared eradicated in 2011, marking it as one of only two viral diseases eradicated globally, the other being the smallpox. And the one more famous, the animal infectious disease, the canine distemper, the CDV. The CDV affects domestic and wild carnivores, especially dogs, wolves, and foxes. It causes the respiratory, gastrointestinal, and neurological symptoms and can be fatal. Has a broad host range, impacting species beyond domestic animals, and vaccination remains essential to control its spread among pets and wildlife. Still, our pets are vaccinated against the canine distemper virus. And the Bastidipity ruminants virus again affects small ruminants like sheep and goats, causing PPR, also known as goat plague, marked by fever, sores in the mouth. That is why they got starvation and the diarrhea, malnutrition, and respiratory symptoms, and it can be fatal, particularly in young animals. The vaccination campaign have been implemented to control and potentially eradicate PPR. And one more thing is cetacean morbilliviruses. The cetacean means the animals living in ocean. The CMV affects marine mammals, particularly dolphins, whales, and porpoises. The infected animals can suffer from respiratory, neurological. Neurological is really like a measles in human beings, and the immune system issues, and leading to high mortality in some cases. Has been associated with significant die-offs in various cetacean populations around the world. These viruses highlight the morbillivirus genus's impact on both human and animal health, spanning a wide range of animals. And efforts like vaccination have played a crucial role in control of this disease and even eradicating some of these viruses. Um, I'd like to deal with the persistence to physical and chemical action against the viruses because everybody, almost everybody got interest in how much they can survive in environment or in our body, this virus. The temperature, the first is the temperature, the half-life calculation of 3 hours at 37 degrees Celsius or 2.2 minutes at 56 degrees Celsius. As you knew, the 56 degrees Celsius is the serum sample inactivation degree, right? And the pH, the stable between the pH 5 and 10, 5 between 5 and 10, and thus inactivation at the very basic pH 11. And the disinfectants and the chemicals, the effective agents include alcohol, the ether, because they have an envelope. The envelope they consist of the lipids. The lipids is very weak against the alcohol and ether, even like the organic material, and a common detergents and is susceptible to most disinfectants like a phenol, the sodium hydroxide, the 2% and 24 hours. Survivals. They survives for long periods in chilled and frozen tissues. Epidemiology is like that. The Bastidipity ruminant represents one of the most economically important animal diseases in areas that rely on small ruminants as a way of making a living. And the morbidity rate in susceptible populations can reach 90 to 100%. Morbidity is really skyrocketing, and the mortality rates vary among susceptible animals, but can reach 50 to 100% in more severe instances. Morbidity high and mortality high. Both morbidity and mortality rates are lower in endemic areas, and in adult animals when compared to young. The lower in endemic areas, in other words, the very high can be high in first outbreak the countries. Host accessibility. The primary hosts are domestic small ruminants, sheep and goats. The PPRV infection in camels is continuously being reported from the endemic regions, and wild ruminants, the gazelle and the Siberian ibex and impala are susceptible as well. The cattle are usually infected asymptomatically and are not known to transmit the disease to other animals. The usual hosts, such as pigs, are also considered as susceptible to PPRB infection. Yeah, this is new information maybe for you. The unusual hosts, the pigs can be infected. The PPRB presence in wildlife, the many wildlife, the Europe and the South Africa and the Asia area. The PPR status is PPR free suspended, and that check this area is a PPR free suspended, and this is a PPR free so far, and the gray one, the PPR present. The African, the most African, the Middle East, the Southeast, and Mongolia, China, the very susceptible, and the PPR present. In Korea, not yet outbreak. I'd say not yet outbreak, but that maybe in the future, anytime soon, can be affected by this virus, unfortunately. The transmission mainly by aerosols or direct contact between animals living in close quarters. The virus is excreted in bodily fluids of infected animals, especially secretions from eyes, nose, mouth, the feces. The fomites may be the means of spreading infection, bedding, feet, and pasture, and water flows. The no carrier state. The no carrier state. So got infected, they present the clinical signs. The seasonal variations, more frequent outbreaks are during the rainy season or the dry cold season, probably associated with seasonal periods of increased local movement and trade in goats. If there's a human activity, is it in relation to the human activity, the seasonal variations can happen. The distribution is the first reported in 1942 in Ivory Coast, this Ivory Coast, maybe this area, and it spread far beyond its origin in Western Africa. And in the past 15 years, its dissemination has been exponential, very contagious. The PPR is now present in over 70 countries across Asia, Africa, and the Near and Middle East, having reached Europe in 2016, Georgia. Yeah, very hyponencially spreading. Likewise, no number of years of PPR presence in 2005, since 2005 to July 2018. The color is darker and darker. The PPR, its meaning is the PPR is very contagious and spreading quickly, exponentially. Still distribution. It has the four lineages. The lineages 1 to 3 have been found in Africa. Lineage 4 was mainly restricted to the Middle East and Asia, with few exceptions of lineage 3 being reported in Yemen and Oman, and mixed lineages of lineage 3 and 4 in the United Arab Emirates and Qatar. However, lineage 4 has now established its presence all across the PPR endemic areas with frequent outbreaks in Africa. Clinical signs, the acute form is a fever, definitely. And eye and nasal discharge, and erosive lesions in the oral cavity, the severe watery blood-stained diarrhea, listlessness, because they very sick. They are very sick. That is why they seem to be very listlessness. And the respiratory signs, the coughing and pneumonia, and abortion may occur, the death with case fatality rates as high as 90%, though more commonly around 20%. The PPRV also causes immunosuppression. Yeah, uh, it deserves it deserves taking our eyes on this. It deserves to be focused, immunosuppression. The clinical signs can be divided to the proacute form and the subacute form. The proacute form is frequent in goats, especially situations of immune-naive introductions into instances of circulating PPRV, and a high fever, depression, and death, the higher mortality. And the subacute, in case of subacute form, the frequent in some areas because of local breed susceptibility, and usually 10 to 15 days development with inconsistent signs. On the 60th day post-infection, the fever and the serous nasal discharge is observed. The subacute form is all the time problematic because it takes time to present the clinical signs. So we don't know during that time, that period, we don't know that those animals got infected by the PPRV. And the fever falls with the onset of diarrhea. Yeah, diarrhea happens and the fever just dropped. It reasonable. And if it is severe, then may result in dehydration severely. Diagnosis. The incubation period is typically the 4 to 6 days, but may range from the 3 to 10 days. In most cases, clinical signs appear in 3 to 6 days. For the purpose of the OIE Terrestrial Animal Health Code, the incubation period for the PPR is 21 days. Very long. For the laboratory diagnosis, the sample should be prepared in the live animals, the swab, the conjunctival discharge, and from the nasal and the buccal mucosa. For virus isolation, the whole blood collected in EDTA. And for serologic needs, a clotted blood can be collected. Keep in mind, for virus isolation, the whole blood should be collected in EDTA. And post-mortem, the lymph nodes, the spleen, lungs, small intestine can be good samples. For the laboratory diagnosis, the procedure is as follows. Identification of the agent is conducted by the nucleic acid detection and identification RT-qPCR, the immunocapture enzyme like the immunosorbent assay, the ELISA, and the culture and isolation methods is the very critical method to confirm what the virus is infected. And the aggregate immunodiffusion and then the counter immunoelectrophoresis. And for the serological test and the virus neutralization and a competitive enzyme-linked immunosorbent assay. The gross pathology is those lesions. The lesions associated with the PPR, they're very similar to those observed in cattle affected with the rinderpest. So we have some knowledges from the experiences with the rinderpest. They accept the prominent crusty scabs along the outer lips and the severe interstitial pneumonia frequently occur with the PPR. They keep in mind that there definitely are the respiratory. They're starting with the upper respiratory symptoms and the deep in respiratory symptoms like pneumonia. The emaciation and the conjunctivitis and erosive stomatitis. Lesions on the hard palate and the pharynx and upper third of the esophagus. The rumen, reticulum, omasum rarely have lesions. So yeah, to keep in mind, in the rumen and reticulum, the 1, 2, 3, third, the stomach doesn't have the lesions. The small streaks of hemorrhages in the first portion of the duodenum and the terminal ileum. The necrotic or hemorrhagic enteritis with extensive necrosis, so sometimes severe ulceration of Peyer's patches. It makes sense because I just say before this is immunosuppressive agent, so that is why it just destroy the make a lesion in the Peyer's patches, the immunoid organ. The congestion around the ileocecal valve at the cecocolic junction and in the rectum, we call that the zebra stripes of congestion in the posterior part of the colon, the small erosions and petechia on the nasal mucosa, the larynx and trachea. And the bronchopneumonia is a constant lesion. All the time we can find the bronchopneumonia inside of lungs, the congestion and enlargement of spleen and liver. It can be said as the maybe this virus is some make some lesion in endothelium or the blood vessels. So that is why the hemorrhage happens. Once the hemorrhage happens, those hemorrhaged blood go to the spleen and liver. The congestion, enlargement, and edema of most of the lymph nodes. Gross pathology, the PPI viruses, the necrotic lesions, They hear the tonsils and necrotic lesions. This is a zebra stripping. The ileocecal junction usually can have this kind of a zebra stripping. The large and they continue to the large intestine and consolidate the lungs because it has a respiratory symptoms. That means the virus likes the respiratory organs. So the consolidation that can be appeared in the lungs. The histopathology, the common characteristics of a morbillivirus infection, the syncytial formation, and extensive necrosis. Syncytial, the bigger cell, that means the syncytial formation. Again, the histopathology, the cells necrosis and the syncytial cells can be appeared. Again, the gross pathology, ulcerative glossitis, the tongue lesion, the tongue of goat. There are multifocal erosions and ulcerations. It can be changed to the scabs. And the ulcerative stomatitis, it means it is destroyed here. And the arrows are necrotic and then syncytial cells appeared. This is again you have a see here. This area is a necrotic areas, and then this area is the retropharyngeal lymph nodes. So a moderate depletion here. The moderate depletion of lymphocytes and lymphoid follicles are multifocally poorly distinct or absent. This is ileum, the moderate depletion of Peyer's patches, and the villi is missing. Okay. The immunohistochemistry, the PPRV appeared in immune organs. This is lymph nodes or lymphoid organs. Lymphoid organs or lymph nodes, the virus appeared. It means the virus likes the immune organs and maybe it can be said the macrophages or dendritic cells phagocytosed and then delivered to the lymph nodes. And we can think that in two ways. Paracortical areas of lymph nodes and the diffuse lymphoid tissues of tonsils affected most significantly. Okay, again. And then I'd like to say about the differential diagnosis. The differential diagnosis, so-called DDX, is important to distinguish the PPRV from other disease. The contagious capillary pleural pneumonia is a famous disease in goats. And the blue tongue should be definitely should be distinguished from this disease. And the pasteurellosis also may occur as secondary infection to PPR. Actually, the PPRV, once they destroy the barrier of our body, and then the Pasteurella multocida, the bacteria, can be infected secondarily. And contagious ecthyma, the contagious ecthyma's clinical sign is very similar. So very carefully we have to distinguish. And the foot and mouth disease, and hot water, and coccidiosis, Nairobi strip disease, and the mineral poisoning. The mineral poisoning is also we should focus on the mineral poisoning because they are grazing outside. The ones that they go out and grazing activity, somehow those lands can be damaged by the heavy metals. So maybe the human construction thing, the construction activity can leave the heavy metals. So they can be affected the mineral poisoning as well. So we have to think the mineral poisoning as well. No specific treatment. Yeah, no specific treatment. But in endemic areas, the supportive care that were known. And the supportive care and treatment of a bacteria and a parasitic co-infections may decrease mortality. And antibiotics for the secondary infection may prevent the secondary pulmonary infections. The prevention is the global rinderpest eradication program showed that using an effective rinderpest vaccine that protects against all virus strains was crucial to its success. So we can have some lesson from the global rinderpest eradication program. Similarly, the efficient PPR vaccines are available and can induce lifelong protective immunity in vaccinated animals, unfortunately. And the vaccination is thus one of the key tools to controlling PPR. The OIE, the OA, and the FAO, in their joint strategy for control and eradication of PPR, have set the goal of eradicating the disease by the 2030. The FAO has a very deep interest on the eradication of the PPR because the Mongolia, for example, the Mongolia has a they are very popularly eat the goat meat or the sheep meat, lamb. So the PPR should be first of all, the PPR was treated as a top priority. The regional roadmap is like that. The affected countries have been grouped in groups into 9 regions. The regional action plans outline the control and eradication measures to be implemented at national and regional level. The PPR West Eurasia roadmap and the PPR East Asia roadmap, the South Asia, the roadmap, those continents are divided into the various areas and they run the eradication roadmaps. And there are some medical prophylaxis. Since the global eradication of rinderpest, heterologous vaccines should not be used to protect against the PPR. So there's several homologous PPR vaccines are available, being cell culture attenuated with strains of natural PPRV. In 1998, the OIE World Assembly endorsed the use of such a vaccine in countries that have decided to follow the OIE pathway for epidemiological surveillance for within the past in order to avoid the confusion when serological surveys are performed. These live attenuated PPRV vaccines are commercially available. Maybe in somehow in the future we we should use those vaccines maybe. And the UA PPR Regional Vaccine Bank, the PPR Vaccine Bank mechanism allows high quality vaccines complying with OA international standards to be delivered to countries in a timely manner. As of June 2016, the OI has already delivered a total of the more than 14 million doses of PPR vaccines to targeted African countries. The OI is currently strengthening and expanding its PPR vaccine bank in order to continue to provide its member countries with access to high quality vaccines at a highly competitive rate in the future. The PPR free countries here is Korea, South Korea. See here. A lot of affected neighbor countries got in the countries and zone without the official status of PPR. Even so, Korea still has the free status. But in any time soon, the Korea can be affected. But this is not. This is maybe I'd say this can be a natural thing. But so far, the people in relation to the animal disease really got effort on protection of our animals inside of South Korea very well. Okay, and I'd like to quickly introduce the vascular stomatitis as of my presentation, the last of my presentation. The vascular stomatitis is a contagious livestock disease caused by the New Jersey virus or the vascular stomatitis Indiana virus, the two of which are called the vascular stomatitis virus. We call that the VSV. These belong to the genus Vesiculovirus, vesicle-making virus, the Rhabdoviridae, the family, the vector-borne and the through blood-feeding or tissue-feeding flies, most notably sandflies, the culicoid biting midges, and the black flies. Incubation period of 2 to 8 days in the vesicles develop after 24 hours, very quickly, the vesicle made. The mainly affects the horses and cattle. The most susceptible the species is horses, can affect swine, but can affect, in fact, swine, sheep, goats, llama, the alpaca, and even people, causing the flu-like symptoms. Okay, simply I introduced the vascular stomatitis, and I think I have to wrap up these presentations. Thank you for listening. Thank you.